Bueno, ya que me han dado la oportunidad de estar aquí presentando a un antiguo compañero mío al que admiro, en primer lugar quisiera, ya que estoy aquí, aprovechar y agradecer y dar mi sincera felicitación al comité de organización de este congreso por su esfuerzo y por el tiempo invertido en su preparación. La verdad que estoy viendo a todos y bueno, es cierto que han gastado muchísimo tiempo y están muy preocupados porque todo salga bien. Entonces, me gustaría felicitarles y agradecerles todo este esfuerzo. Y bueno, también quisiera agradecerles el permitir moderar esta mesa y presentar al próximo ponente. Bueno, tenemos el honor de contar con la presencia de un excelente investigador del ámbito de la biolingüística, al que tuve el placer de conocer en la Universidad de Huelva y al que sigo con gran interés, el profesor don Antonio Benítez Burraco. Este profesor es doctor en Lingüística por la Universidad de Oviedo y doctor en Bioquímica por la Universidad de Córdoba. Además, es licenciado en Filología Eslava por la Universidad de Granada y licenciado en Biología por la Universidad de Sevilla, universidad en la que actualmente desempeña su actividad docente e investigadora. Es miembro del grupo de investigación Biolingüística, Fundamentos Genéticos, Desarrollo y Evolución del Lenguaje. Y su labor investigadora se centra en el análisis de los fundamentos biológicos del lenguaje y de su evolución en la especie. Se trata de un investigador incansable y muy productivo. Ha publicado alrededor de 100 artículos de investigación, 15 capítulos de libro, 2 libros sobre este tipo de temas, además. Cuenta con unas 85 participaciones en congresos nacionales e internacionales y ha impartido alrededor de 50 conferencias invitadas, como la de hoy, lo cual no es poco. Entre sus publicaciones, citar algunas, quizá las más recientes, o las que señala él, The Oscillopathic Nature of Language Deficit in Autism, o A Bilinguistic Approach to Language Disorder, Towards a Paradigm Shift in Clinical Linguistics, perdón por mi inglés, Osteogenesis and Neurogenesis, a Robot's Link Also Language Evolution, o El origen del lenguaje, o Language Impairment in a Case of a Complex Chromosomal Rearrangement with a Breakdown Downstream to FOXP2. Además, ha participado en numerosos proyectos de investigación. Actualmente desarrolla la globularización y trastornos del cerebro aptos para el lenguaje. Bueno, este es el reciente, sin embargo tiene otros, por ejemplo, los trastornos del lenguaje como celopatía, hace un cambio de paradigma en la lingüística clínica, o Towards a Comparative Biolinguistics. Se trata, sin duda, de un referente en los estudios de biolingüística, genética, lingüística clínica y neurolingüística, tanto a nivel nacional como internacional. Mi mayor y sincera admiración para este investigador. Estoy seguro de que nos va a enriquecer y nos va a hacer disfrutar con su conferencia titulada The Golden Mean, Assistant Biology Approach to Language Disorder. Así que, sin más preámbulos, le damos la palabra.
Bueno, muchas gracias, Mario, por tus palabras. Muchas gracias a todos por estar aquí, a los organizadores del Congreso por invitarme. Y, bueno, yo voy a leer mi conferencia en inglés, por un poco de deferencia a los invitados que vienen de fuera, pero luego estoy encantado de responder a vuestras preguntas en inglés o en español, como veáis, ¿no? So, I wish to start by sincerely thanking the organizers of the 5th International Congress of Clinical Linguistics, and particularly to Professor Carmen Baro and María Jesús Paredes and Miguel Casas, for kindly inviting me to present this plenary talk at this session on genetics and language. And of course, to all people in the room for being here. In my talk, I will discuss about new theoretical approaches to the nature of language disorders that account for many of recent findings about their biological nature in the domain of genetics, brain physiology, or behavior. Hopefully, these new models will help physicians, speech therapists, and clinical linguists to better interpret the relevance of these findings, improve their understanding of the etiology and symptomatology of disorders, and ultimately achieve earlier and more confident diagnosis of language disorders, as well as more efficient therapies. On paper, clinical categories like dyslexia or specific language impairment refer to cognitive disorders in which only language is impaired and that can be differentiated from other similar categories at all levels: linguistic, cognitive, neurobiological, and genetic. However, Things are usually less clear-cut and more difficult to handle, essentially because the boundaries between disorders are blurred at all those levels. This circumstance is expected to impact negatively on the diagnosis and the therapeutic approaches aimed to ameliorate the symptoms and deficits associated to these conditions. This problem is not easy to fix. My take-home lesson today will be that clinical linguistics will benefit from a shift of focus in the line of the ongoing evo-devo revolution in biolinguistics and more generally in biology. Instead of relying on the analysis of the phenotype in the adult state, more attention should be paid to developmental dynamics in pathological populations across all levels of biological complexity, from genes to language deficits. Importantly, as I will try to show, this should allow to find more reliable endophenotypes of these conditions, that is, disorder-specific biological markers of the disease. Today, I will highlight brain oscillations as the most promising of such endophenotypes. For better achieving my goal today, I have structured my talk as follows. First, I will provide a brief characterization of the messy scenario for clinical linguistics, as I sketched it before. I will stress that this messiness has increased with the advent of the omics revolution in biology. Afterwards, I will present several reasons for adopting an eco-evo-devo approach to language disorders, which with its heavily reliance on concepts like canalization, developmental plasticity, robustness, evolvability, and adaptive landscapes. In the third part of my talk, I will focus on brain rhythms. I will defend that brain oscillation could be the best bridge between gene mutations at the bottom and language deficit at the surface, and thus the most reliable endophenotype of language disorder. I will finish with some conclusions and advancing new avenues of research for the future. As I said in my introductory remarks, things for clinical linguists and speech therapists are not usually crystal clear. To begin with, patients commonly show symptoms that are compatible with more than one disorder, linguistic or not linguistic by nature, to the extent that comorbidity is a frequent outcome of clinical practice. At the same time, people suffering from a particular disorder usually exhibit linguistic disabilities that are pretty variable. In order to apprehend this variability, different subtypes of the same disorder are usually posited, in which one aspect of language is claimed to be more impaired than others. Importantly, problems with language at the surface, so to speak, are only indirectly related to the cognitive deficits at the bottom. This seemingly contributes to increase the variability of the symptoms and makes the categorization of disorders more troublesome. On the whole, it seems that different disorders or different subtypes of the same disorder can emerge from the same broad cognitive deficit, which can appear differentially in different populations and or environments. Hence, the alleged heterogeneity and or comorbidity. In other circumstances, however, different deficits, specific or not to language, can contribute to the same disorder. If they result in a common symptomatic profile, it is then possible that in each subtype of a particular disorder, a different deficit prevails. At the same time, in other populations, any of these underlying deficits can result in a different disorder. Hence, the purported heterogeneity and comorbidity. Since another important concern is that very frequently problems with language in the affected people concern to quite broad aspects of language, to the extent that they do not match units, levels, features, or operations that are important for modern linguistic theory. As a consequence, clinical typologies are sometimes weird for linguists. For instance, according to some accounts, there exists a syntactic pragmatic subtype of specific language impairment, But we linguists carefully differentiate between the knowledge of a language, that is the grammar, and how this knowledge is put into use, that is pragmatics. Actually, for most linguists, for most neurolinguists indeed, pragmatics involves many other things beside our linguistic competence. Finally, consider that the clinical profile of the patients usually change throughout development to the extent that the affected subjects can switch from one subtype to another of the same disorder as they grow. Accordingly, one cannot simply assume that deficits in the adult state are the same as those apparent in the infant state, and vice versa. Comorbidity, heterogeneity, and variability are observed as well at the neurobiological level. Accordingly, the brain areas found affected in one disorder can be found impaired in people suffering from a different disorder. Moreover, it's frequently observed that the affected regions give rise to mixed symptoms. Overall, it's not clear whether the involved regions are multifunctional by nature or perform instead some basic computations that are required for language and for other cognitive abilities. Lastly, it commonly happens that the limits of the affected areas are differently placed in different subjects. Things are not easy to interpret at the molecular level. Different candidate genes and risk factors for different language disorders have been identified to date. However, it's not one but many genes that usually contribute to each disorder. Typically, several pathogenic variants of each candidate gene have been identified. At the same time, other variants of the same gene have been proved to contribute to the language abilities of the neurotypical population. Importantly, the same mutation in the same gene can cause the disorder in some individuals, but not in others. Conversely, pathogenic variants of a particular candidate gene are frequently absent in people affected by the disorder. What is more, the same mutation in the same gene can give rise to different disorders in different subjects, to the extent that candidate genes for a particular disorder are frequently mentioned as candidates for several other clinical conditions. Finally, it's frequently observed that mutations in genes encoding proteins that are functional related, for instance, if one regulates the expression of the gene encoding the other, can give rise to different disorders in different people or in different environments. As I noted before, the advent of the so-called omics revolution in biology has turned this complex scenario even more complex. On the one hand, the amount of biological data about disorders has grown exponentially. On the other hand, we have learned that there are additional levels of complexity that have to be explored if we really want to gain an accurate picture of the real nature of disorders. Accordingly, epigenetic change, modifications in protein networks, alterations of signaling pathways, abnormal patterns of neural assembly, or aberrant patterns of neural synchronization need to be considered on a par to gene mutations, abnormal neuroimaging results, or language deficits. Importantly, as I will try to show in the last part of my talk, when I discuss about brain oscillations, it might well be that the specificity of language in cognition, and hence the idiosyncrasy of language disorders that we have been unable to find at the genetic or the symptomatic levels, can be found instead at some of these new levels of biological complexity. Overall, it seems that clinical linguistics, which has traditionally focused on the cognitive and linguistic evaluation of patients and the analysis of corpora of disordered language, face today to a triple challenge. First, it needs to consider new types of evidence as provided by many different areas of research interested in language disorders. This is the challenge of multidisciplinarity. Second, it needs to incorporate new methodologies and tools, which are often complicated to understand and to use. This is the challenge of technification. And third, it needs to formulate new models of language disorders that take into account this increasing body of data about the biology of language disorders, but particularly fresh models of development and evolution as currently discussed in biological science. This is the challenge of theorization. Today, I will mostly focus on this last challenge. I believe that this will be possible only if a systems biology approach to language disorders is adopted. This approach is aimed to characterize from a holistic perspective the complex interactions among myriads of biological components that take place within the brain of people with disorders when processing language, as well as the emergent properties resulting from such interactions. Among other things, this fresh approach is expected to circumvent the caveats and shortcomings of current typologies of language disorders, which are mostly based on symptomatic criteria or an etiological criteria. Typologies based on symptoms often fail to categorize and characterize patients unambiguously, essentially because of the widespread problem of comorbidity and heterogeneity discussed before. Also, they usually fail to incorporate etiological factors in a disorder-specific way, particularly what we have learned in the last decades or years about the genetic foundations of neurodevelopmental conditions. For instance, as far as genes are concerned, it seems now that language disorders do not result from the alteration of one or a few disorder-specific genes, but of many, if not most, of the genes expressed in the brain. Accordingly, it seems better and more biologically sounded to characterize each disorder in terms of whole brain transcriptomic profiles that we expect to be disorder-specific. But this could be not enough, considering that etiological classification of disorders usually fail to incorporate many of the other involved factors, like epigenetic factors. More importantly, they fail to explain why only a bunch of distinctive symptomatic profiles result from the interaction of thousands of potential etiological factors, some of them altered and some of them intact. In the picture, schizophrenia and autism, with most, if not all, of these pieces being shared across disorders that exhibit different symptomatic profiles. Actually, this messy scenario, as I call it at the beginning of my talk, is easy to interpret if we rely on eco-evo-devo theories in biology, which build on the deep link between the environment, development, and evolution. According to this approach, language evolved as a result of a specific change in the developmental path of the hominin brain in response to change in the environment in which our ancestors lived. And it is the most recently evolved components of human cognition that are expected to be the most sensitive to the deleterious effects of developmental perturbations resulting from environmental change, either internal to the organism or external to it. This circumstance seemingly explains as well two widespread outcomes of clinical linguistics. First, although as note, the etiology of disorder is quite diverse, some deficits are shared by nearly all conditions, and they usually concern to morphophonology and to highly demanding computational tasks like agreement. Second, although as also note, the number of etiological factors is high, the number of disorders is far smaller. Under this eco-evo-devo view, these two outcomes are not surprising. Regarding the first finding, language evolution can be also viewed as the emergence of new interface between basic cognitive blocks that are otherwise very robust after millions of years of stabilizing selection. and as a consequence are not usually found impaired in disorders. By contrast, because of their evolutionary novelty, and ultimately because the less resilience of the networks they rely on, these new interfaces are most sensitive to damage. Regarding the second one, that is, why the number of disorders is far smaller than the number of etiological factors involved, it might be hypothesized that the disorders that are finally attested by clinical linguistics might be the only possible set of phenotypes resulting from the interaction of the diverse factors, normal or abnormal, that regulate the development of the brain. In Evo-Devo theories, the limited set of phenotypes available during development are construed as definite areas within a morphospace or adaptive landscape. Putting it differently, developmental dynamics canalize development towards a restricted set of ontogenetic paths. Accordingly, what we call the neurotypical language brain might be viewed as the outcome of a successful canalization of the otherwise widespread developmental noise in the form of gene mutations, minor brain anomalies, and the like. In turn, what we call language disorders can be viewed as suboptimal canalizations of more severe developmental disturbances, deleterious gene mutation, widespread brain damage, and the like. What we need to determine are the best parameters defining the language morphospace. Consider as a good example the shells of ammonites and nautili. Similarly to language disorders, the number of different shell forms was small: coil, uncoil, and helical. Nonetheless, the shell morphology depends on two parameters: rib expansion rate and rib coiling tightness, which change continuously, similarly to what happens with the etiological factors of disorders. As far as language disorders are concerned, one possibility might be relying on aberrant gene expression profiles in the brain, with the aim of characterizing at the molecular level the diverse states attracted through development. Whereas some expression profiles could characterize language disorders, other, or perhaps several ones, would be typically associated to the intact faculty of language. Nonetheless, in our recent research, we are checking the hypothesis that brain rhythms might be a better or perhaps the best candidate for properly defining the morphospace of language growth in the species, either pathological or neurotypical. Several reasons justify our current interest in brain oscillations. First, they are primitive components of brain function. Second, we expect them to be connected to some computational primitives of language, thus allowing to understand and not just to localize brain functions. As famously noted by David Popel, current neurolinguistic studies suffer from two crucial shortcomings. On the one hand, they make two broad conceptual distinctions, like syntax versus semantics, morphology versus syntax, and the like, which involve several different neural components, computations, and representations. On the other hand, the fundamental elements of linguistic theory cannot be reduced or matched up with the fundamental biological units identified by neuroscience. It is then urgent to spell language in computational terms that are at the appropriate level of abstraction, that is, that can be performed by specific neural populations. We regard brain rhythmicity one and perhaps the most suitable of these appropriate levels of abstraction. For example, the assignment of language-relevant features like tense of case can be satisfactorily interpreted as the embedding of high-frequency oscillations inside oscillation operating at slower frequency. Similarly, some rhythmic features of speech have been successfully related to specific brain oscillations. A third reason is that the hierarchy of brain oscillation has remained remarkably preserved within mammals during evolution. Not surprisingly, the human-specific pattern of brain activity underlying language processing can be evolutionarily linked to the oscillatory signature of the primate brain. My colleague and co-author Elliot Murphy discussed in a recent paper the emergence of the human language oscillome, that is, the language-related phasal and cross-frequency coupling properties of neural oscillations. And he argues that the human language oscillome simply reshape the ancient primate oscillome we inherited, although the reverse also happened, and this is why we find in human languages that semantics impose particular constraints on possible syntactic structures. A final but very important reason is that each cognitive disorder can be associated to specific profiles of brain rhythmicity. This is of particular interest for clinical linguistics, because brain rhythms are highly quantifiable and heritable traits. They might result in robust biomarkers or endophenotypes of language disorders. For example, the gamma band dysfunction has been hypothesized to be a robust biomarker of autism spectrum disorders. Nonetheless, if we really want to use these oscillopathic profiles as reliable biomarkers or endophenotypes of language disorders, we first need to map these abnormal disorder-specific patterns of brain oscillation to the language deficit that are also typical of each disorder. Pretty obviously, this translational effort should allow to gain a better understanding of the nature of the disorders and consequently the etiology of the language deficits associated to each condition, particularly of the effect of gene mutations. In our recent work, we have proven that this translational effort is feasible. We have shown that the language deficit observed in conditions like autism, schizophrenia, or dyslexia can be explained in terms of aberrant change in the normal oscillatory activity of the brain. For example, when processing speech, gamma oscillations correspond to phonetic features and are important as well for accessing stored templates from memory. As a consequence, degraded gamma and zeta synergy in autism may explain the problems that people with this condition experience with speech perception, tone recognition, and parsing phonemic representation. Even more importantly, we have shown that some candidate genes for these conditions are related to brain rhythms. In some cases, it is even possible to draw bridging links between all the involved levels of biological complexity, from gene mutations to abnormal brain oscillation to language deficits. For example, this gene is ZNF8 and the like, encodes thin finger binding protein and is highly expressed in the hippocampus and the neocortex, particularly during late embryonic development. The hippocampus is a source of theta bands which play a major role in coordinating distributed cross-cortical activity, in particular activity in the prefrontal cortex. Because of their involvement in working memory as a filter that impose memory-related rules, and because of the overall role of the hippocampus in the transformation of individual experience into semantic structures, such as maps of schemas, hippocampal theta are expected to explain core aspects of language processing, both syntactic, like the chunking of syntactic objects, and semantic, like category fluency. Pathogenic polymorphism of this gene have been related to semantic problems in subjects with schizophrenia. Accordingly, significant decreases in the coactivation of the right hippocampus within the whole hippocampal network, as well as in intrahippocampal theta band, have been found in risk homozygotes for one variant of the gene. Interestingly, people with this risk allele show a greater coactivation of the hippocampus and the prefrontal cortex, specifically the suprafrontal gyrus. Likewise, several polymorphisms of this gene have been associated to verbal deficits in people with autism, who exhibit a reduced expression of this gene in several brain areas. Just to put another example, GRIN2A is a gene that encodes the subunit 2A of the MMDA receptor, which plays a key role in long-term potentiation, important for memory formation and learning. This function similarly results in part from its regulation of gamma oscillation formation and modulation. Mutations in this gene are found in people suffering from different forms of epilepsy aphasias like Rolandic epilepsy or Landau-Kleffner syndrome. Specifically, they are associated with errors in articulation and with problems with pitch and prosody, which pertain to the syntax-phonology interface. In turn, this deficit can be tracked to an abnormal gamma activity involved in the processing of fast-rate phonemic and syllabic information. Interestingly, most of these candidates for language disorders that play a role in brain rhythmicity are functionally interrelated and map onto particular regulatory pathways, cell types, or functions, as well as aspects of brain development and function of real relevance for language processing, particularly through dopaminergic, GABAergic, and glutamatergic synapses. Importantly, too, these genes exhibit a distinctive disorder-specific pattern of up and down regulation in the brain of patients, as shown here by the expression grids of this gene across the brain regions profiled by the Allen Brain Atlas. This contributes to bridge genes to oscillations to language features. Notice as well that the specificity of the molecular signature of each disorder relies not on the set of genes involved, which are essentially the same, but on their expression patterns in each brain region, which is different in each condition. Finally, and perhaps not surprisingly, it is important to note that several of these genes that are responsible for basic aspects of the oscillatory activity of the brain relevant for language show difference in their methylation status between Neanderthals and modern humans. It is obvious that we cannot track the oscillatory activity of the brain of extinct hominins, but as I showed before, from these molecular differences that are suggestive of differences in the expression pattern of these genes, we can infer differences in cognitive functions important for language. Overall, because of its bridging role between the genes at the bottom and language feature at the surface, both developmentally and evolutionarily, brain rhythms could be the biological level at which the specificity of each disorder emerges. The golden mean, just to say. Let me add a final note to this vindication of a systems biology approach to language disorders, or an eco-devo clinical linguistics, which is quite the same. As is pretty obvious, organisms are not isolated entities. Instead, they are open systems in close contact with the environment. Years of research on child language, either neurotypical or impaired, have demonstrated that language development is heavily influenced by external factors that can contribute very significantly to the emergence of language disorders. Systems biology is aimed as well to capture the effect of these interactions. This is the eco side of eco-evo-devo. To put just one example, our microbiota, and more generally the gut-brain crosstalk, has been found to control important aspects of brain development and function. Increasing evidence suggests that alterations of the gut microbiota axis can disturb some of the cognitive networks encompassing emotional and social domains in neurodevelopmental disorders, and accordingly contribute to the social and emotional deficits observed in conditions like autism and schizophrenia. It is true that mechanistic insights are still pending, but as noted by the authors of this paper, a system-level dimensional approach to this issue can result in findings of clinical relevance. I think that the same can be said of language disorders. This is not surprising, after all, because the environment has an impact on all the levels of biological complexity related to language impairment and to brain oscillations, of course. Accordingly, we should expect a connection between environmental cues, abnormal brain development, altered brain oscillation, and language deficits. It's pretty obvious that this research has a practical side too. If we succeed in this translation of language dysfunction into disorder-specific patterns of brain abnormal oscillations, we might be able to diagnose language disorder earlier and in more accurate ways. In my opinion, several complementary lines of future research are of particular interest for improving this system biology approach to language disorders that heavily builds on brain rhythms. First, we need to disentangle the molecular mechanisms that channel or fail to channel variation at all levels of biological complexity. Second, we need to improve eco-Devo-friendly depictions of the modularization of the disordered brain. Third, we should optimize our current models of the linguistic ontogeny in people with disorders. And finally, we should pay attention to emergent properties and to properties that fail to emerge, because language is undoubtedly a complex system, and many of the properties of complex systems are emergent by nature. Summarizing what I have argued today, I think that characterization and classifications of language disorders based on intermediate level components, and particularly on the coupling between brain oscillations during language processing, are more biologically motivated and more theoretically grounded than others, particularly the ones currently used by clinical linguists, either symptoms or causes. Accordingly, they are expected to help us identify more robust endophenotypes of language disorders that can be used as reliable hallmarks for an earlier and more accurate diagnosis of these conditions. And that's all. Thank you very much for your kind attention, and I will be glad to answer your questions.
Okay, thank you very much for your interesting conference. Now it's time for question. So, question that you may have. Question, también podéis preguntar en español si queréis. Hugo.
Bueno, muchas gracias, Antonio, por esta conferencia tan futurista, pero que ya está aquí con todas esas publicaciones que has mencionado. Te quería preguntar, en cuanto a los ritmos cerebrales, ¿no se plantea también un problema? ¿O qué conocimiento hay respecto a la ontogénesis de los propios ritmos cerebrales?
¿Qué problema hay? ¿Por qué? ¿En qué sentido?
No,
que si
pregunto,
cambian...
¿qué conocimiento hay? Porque al final seguimos teniendo, entiendo yo, el problema de explicar la ontogénesis de los propios ritmos, ¿no?
Sí.
Como reflejo de los procesos.
Sí, bueno, pero es lo mismo que se observa con la neuroimagen tradicional, ¿no? Que la activación de las áreas va cambiando de localización ante el mismo tipo de estímulo a lo largo del desarrollo. Entonces, bueno, sería el mismo problema, habría que realmente estudiar bien cómo cambia esa ritmicidad con el tiempo, cómo cambia con el tiempo los trastornos, Pero ya te digo, no sería diferente de lo que, por ejemplo, se hace ahora cuando se trata de localizar el lenguaje. Simplemente la idea es que estos ritmos proporcionan no una localización, sino una explicación del procesamiento.
Sí, yo estoy de acuerdo y estoy al tanto de estas investigaciones.
Pero
y me parecen más potentes en cuanto a la explicación que los métodos tradicionales, pero
sí, claro, necesariamente
no resuelven el problema de explicar la ontogénesis, quería decir.
No, simplemente
nos proporcionan una base, yo creo que mejor, desde luego.
Sí, el reto es un poco el mismo, ¿no? Que no analizar los trastornos al final, sino a lo largo del desarrollo. Pero es cuestión de ir acumulando datos y ir tratando de correlacionar esos patrones de acoplamiento con el proceso de desarrollo del lenguaje, ¿no? Igual que se hace en el estado adulto, pero en diferentes etapas, ¿no? Y terminar el puzzle, ¿no?
Muy bien, gracias.
Está encendido. Muchas gracias, Antonio, por la presentación. No sé si no tiene cabida, no los conoces, los trabajos y el gran proyecto que está desarrollando Annette Karmilov-Smith sobre el tema de cómo es posible que se nazca con predisposición para tener determinados trastornos de la comunicación y cómo esos trastornos se van emergiendo, se van desarrollando. Claro, el planteamiento de Karmilov-Smith no es un planteamiento tan, a ver cómo lo catalogaría yo, tan cerebral como el que has presentado aquí, o tan fisiológico, tan fisicista. Es un planteamiento más mental y más orientado hacia la conducta. Entonces, a mí me gustaría, porque parece que has afirmado que sí los conoces, los trabajos de Karmilov-Smith, me gustaría que, si es posible, trataras de congeniar lo que es la emergencia de tipo mental de estos trastornos con la base cerebral, con la base más fisicista que tú acabas de exponer. Muchas gracias.
Gracias. Sí, bueno, en realidad este modelo es directamente deudor de ese tipo de trabajos, ¿no? Porque implícito a esto está la idea de que la organización del cerebro adulto es un producto del desarrollo. De hecho, ha aparecido muchas veces esta idea de evo-devo, que es la biología evolutiva del desarrollo. Entonces, todas esas ideas están aquí. Aquí lo que se trata un poco es de dar efectivamente un fundamento biológico a esos procesos de emergencia a nivel mental, cognitivo. Pero yo creo que es perfectamente compatible, simplemente estás analizando lo mismo a un nivel distinto. No sé, no quiero decir que no encuentro nada en lo que diga Karmilov-Smith que ella pudiera chirriarle de lo que yo he contado aquí, ¿no? Pero no sé exactamente si respondo a tu pregunta. Claramente, lo que tú observas al nivel que te interesa tiene que estar justificado, tiene que estar explicado por la actividad del cerebro.
A ver, no digo que sea incompatible.
Sí.
Vale, sino que se necesitan establecer rutas que permitan pues una plasmación de lo que ocurre en una base física con lo que emerge funcionalmente de esa base física. Es decir, voy a terminar con esto y tampoco me gusta monopolizar la discusión. Voy a terminar con esto. Todo está en el cerebro, pero no todo es el cerebro. Vale, gracias.
Bueno, sí, se puede ver así, pero yo creo que no todo... en el fondo el cerebro es lo que está procesando los estímulos y dando una respuesta. Entonces, ¿qué es lo que no hay en el cerebro? Quiero decir, pásale el micro.
A mí,
sí, ¿qué es lo que no hay en el cerebro?
Pues yo creo que todo lo que tiene que ver con la conducta no se explica solo por el cerebro. Todo lo que tiene que ver con trastornos que repercuten en nuestro comportamiento no se explica solo por el cerebro. Hay que, dar algún elemento más.
Una cosa es efectivamente, sí, que claramente, por ejemplo, el ambiente tiene un efecto muy importante que no es que en muchos casos va más allá de la predisposición biológica. En eso estoy totalmente de acuerdo, pero al final nuestro comportamiento y nuestras reacciones al entorno se procesan a nivel cerebral. Entonces,
sin duda.
Se pueden analizar a ese nivel, si quieren.
Sin duda, pero mira, por ejemplo, ayer, era ayer o esta mañana, nos presentaban, en algún caso, es que ya no recuerdo si fue una sesión de ayer o de esta mañana, una persona que no tenía ningún tipo de alteración neurológica, sino que simplemente no era capaz de controlar la mente, entonces se ahogaba y no podía hablar. Bueno, pues este tipo de situaciones o este tipo de ejemplos yo creo que solamente tienen explicación si le damos importancia a lo que es el entorno, a lo que son circunstancias personales.
¿Pero qué significa que no tenía ninguna alteración? No tenía ninguna alteración estructural, quiere decir.
Claro, sí.
Pero seguramente funcional, sí. Si tú estudias ese cerebro cuando está...
Pero por eso mismo yo quiero darle entrada a la parte funcional. No quiero monopolizar. Vale.
No, pero quiero decir que...
Lo dejamos así.
Sí.
Yo, vamos a ver, ya termino con esto y no quiero monopolizar. Le voy a pasar el micro.
Así tengo menos preguntas que responder.
Se lo paso a Nuria, que quiere intervenir. Hay un libro reciente de Thomas Fuchs sobre el concepto del cerebro como intermediario. No tanto el cerebro como depósito total, sino como intermediario. Y bueno, yo creo que si uno se acerca a lo que es el estudio de Fuchs, todas estas preguntas que yo estoy planteando, pues a lo mejor tienen...
Yo creo que he sido más radical todavía, ¿no? Cuando, por ejemplo, he puesto esta diapositiva donde en realidad las que controlan nuestro comportamiento son las bacterias que tenemos en el intestino. Eso es más ambiental todavía y menos cerebral, si quieres, ¿no? De hecho, ahí ves la diapositiva, ¿no? Sí. A ti, por las preguntas.
Gracias. Ahora tengo mucha presión.
A ver.
Bueno, muchísimas gracias por la charla. A mí me ha parecido muy interesante y creo que el modelo va a tener mucho desarrollo futuro. Yo quiero hacerte una pregunta desde la más absoluta ignorancia. No sé si sabes, o creo, no sé hasta donde yo sé no se ha hecho, pero a lo mejor sí. ¿Cómo es exactamente esa correspondencia en el modelo entre las frecuencias del habla, que son frecuencias mucho más altas, y las frecuencias de las ondas cerebrales, que son unas ondas de frecuencia mucho más baja? O sea, no sé si se ha descubierto todavía o se va a trabajar en ello sobre cómo es exactamente esa correspondencia.
Pues para eso los trabajos de Poepel y de Giraud
no lo explican. La correspondencia no la explican.
Ya, pero bueno, en eso están, ¿no? Nosotros hemos presentado hoy un trabajo sobre Miguel por ahí está Miguel,
sí,
allí. Sobre precisamente el procesamiento en la dislexia, y él está hablando un poco de eso, ¿no? De cómo las ondas cerebrales van siguiendo un poco, navegando, o como dice Poepel, surfeando, ¿no? Sobre la envoltura acústica.
Sí,
así
pero
que si él quiere...
Eso no es explicar.
Bueno, ya. ¿Qué es explicar? O sea, de momento... yo no he hablado de habla tanto, pero sí de sintaxis, de semántica, y probablemente si hay una explicación causal está ahí, a ese nivel que hemos comentado, ¿no? ¿Qué explicación quieres tú a...?
No sé, yo espero que haya alguna en el futuro, ¿no? Que se desarrolle.
Sí, pero ¿qué problema tienes tú en encontrar que realmente hay un patrón diferente de activación cerebral en respuesta a cada uno de los componentes del habla?
¿Cómo puedes hacer esa correspondencia entre esas distintas frecuencias?
A ver, ¿puedo intervenir?
Sí, claro.
Creo que hay dos formas de ver esto. Una tiene que ver con lo que se sabe sobre las oscilaciones cerebrales, que tienen una funcionalidad, vamos a decir, generalista para diferentes aspectos cognitivos, entre los que se encuentra el lenguaje. Entonces, lo que se observa es que el cerebro, cuando procesa estímulos, se activa a diferentes frecuencias, diferentes grupos de neuronas se activan a diferentes frecuencias, y en este caso, el lenguaje como un estímulo más, las mismas bandas de frecuencia que se activan para procesar un determinado estímulo, que no tiene por qué ser lingüístico, se activan en el procesamiento del lenguaje. Entonces, lo que sí se ha visto es que diferentes componentes lingüísticos que tienen diferentes frecuencias, por ejemplo, transiciones formánticas que ocurren muy rápido en el tiempo, que ritmo silábico, que tiene una frecuencia más baja. Lo que sí se ha observado es que diferentes frecuencias se especializan en el córtex auditivo para procesar estímulos que tienen diferentes frecuencias. Entonces sí hay una cierta correlación entre cuál es el tipo de estímulo que se está percibiendo y qué bandas de frecuencias se encargan, se especializan, por así decir, para procesar en el córtex auditivo concretamente estos estímulos. No sé si esto...
Claro, sí, no, y de hecho es más específico que lo que tú observas en neuroimagen, ¿no? Porque si tú estás haciendo neuroimagen y solo ves activación de una zona concreta del corte, pues todavía menos explicativo que los ritmos. Que no es explicación total, sí, pero es algo más explicativo que lo que era la neuroimagen solamente.
Espero que desarrolléis el modelo, porque yo tengo mucha curiosidad por saber cómo se realiza ese cambio.
Incluso llega un momento en que puede que el procesamiento todavía nos quede mucho por entender, pero si al final encuentras realmente un patrón diferencial para cada trastorno, ya tienes una herramienta en principio de diagnóstico, aunque no consiga realmente explicar en qué, como es una especie de caja negra, pero al menos que funciona, ¿no? Pero es verdad que es interesante, claro, desentrañar lo que hay dentro de esa caja, ¿no?
Solo quiero añadir un poco al hilo de lo que dice Antonio, es que por eso se utiliza el término oscilopatía, en el sentido de que hay un perfil oscilopático diferente para diferentes trastornos del lenguaje. Es decir, no se observan el mismo tipo de anomalías en las bandas de frecuencia para personas con dislexia que para personas con trastorno específico del lenguaje. De ahí el término de oscilopatía aplicada a diferentes trastornos, simplemente.
¿Más preguntas? ¿Questions? ¿Alguna pregunta más? Bueno, pues muchísimas gracias a todos ustedes por vuestra colaboración y agradecerles de nuevo otra vez al invitado su participación. Y nada, es un placer y un honor estar aquí. Bueno, pues ponemos el Congreso hasta esta tarde a las 4:30. Muchísimas gracias. Un aplauso.
Bueno, a ver.
Sí. En lingüística que algo me suene completamente y esto lo consigue que yo me quede.
Bueno, yo intento contarlo
admirado.
sencillito para, bueno, porque de hecho se trata de eso, ¿no? No las cosas estas que no se te cuentan complicadas.